Hi everyone, welcome to this session. So I've been asked to chair a session on the challenges of breeding varieties for regen ag systems. So I'm Dr. Phil Howell. I'm from NIAB in Cambridge. So I'll give a talk. My colleague Stéphanie will give a talk, and Ambrogio will give a talk, and then we'll all be happy to take questions for the last part of the session. So what to expect, as I said, I'm going to talk about sort of mainstream breeding, trials, marketing, why this may not be necessarily ideal for regen ag, and what some of the steps in the right direction are being taken, what needs to change, thoughts about the future. Stéphanie will be focusing mainly on work she's been doing on nitrogen responsiveness. And Ambrogio has a wealth of experience from the Organic Research Centre and some of the work they've been doing, especially in EU projects with partners right across the country and right across Europe. So a different perspective. And as I said, from the end, we'll take questions from the floor and general discussion. So working back from mainstream markets, commercial breeding, obviously, it has to target the varieties that the market wants. The seed royalty market for most of our crops is relatively small and very competitive. There are a lot of breeders fighting for a relatively small royalty market. That means that targeting niches is high risk. Most of the certified seed area is sown with varieties that are on the HDB recommended list. So to move away from those selection criteria, there's an inherent risk in doing that. So by definition, the RL criteria shape the selection strategy earlier in the plant breeders programs. If they're not going to make varieties that suit the recommended list, they're much less likely to take big market share. So it makes sense for breeders to use the same criteria as the recommended list. If these criteria changed, breeders would adapt, and with breeding technologies that are available now, they can actually adapt quite quickly to change to different targets. So their selection criteria further back in their own programs would change. And then this is just some text I took from the RL protocol. So it's about maximizing the yield potential and pulling apart varieties on the basis of yield. So in principle, the husbandry of the trial should be appropriate to achieve highest quality and yield. And then later on, specifically talking about nitrogen. Nitrogen applications should be tailored to give maximum yield within the constraints of obtaining the appropriate grain protein contents. So for wheat, this is talking about either a trial destined for milling quality or biscuit or feed. And for barley, it's tending to talk about malting for brewing or distilling. If we consider testing in alternative situations such as low input or minimum tillage or at one extreme organic, there is work being done by the commercial breeders, but it tends to be deferred until the variety is kind of already finished. So it's later in the near market situation. It's come through the big selection funnel. There's not much variation to play with and make selections from. And just as an aside, often organic growers are looking at older varieties, so some of the old heritage varieties that may have come through almost pre-pesticide testing. So they know that these are varieties that do well without pesticides. These might be varieties that do well in organic situations. So there are other reasons, sometimes to do with marketing, to do with preferring heritage and older varieties. So the principles of mainstream breeding then, as a very lazy phrase, it's cross the best with the best and select the best. It's all about elites pushing yield and disease resistance as much as possible. There's lots of technologies now being brought to bear, so marker-assisted genetic technologies which help with designing the cross in the first place, which parents to put with which parents, and how to select between siblings in the early generations. Again, there are tricks you can pull which can speed up breeding, so you're not just got one cycle in the field every year. You can move things quickly through the greenhouse with some tissue culture work, or even moving whole programs from the northern hemisphere to the southern hemisphere. So these can help, especially with the early generation selection and the multiplication of seed stocks. Fundamentally, field testing normally starts actually in untreated conditions. So the first problems breeders like to solve are disease resistance problems, making sure the crop is the right height, making sure the earliness, the adaptation is correct for the environment we're wishing to grow in. And then mainly that's just because these are things you can do on single plants or very small quantities of seed. Yield, agronomic, predictive quality testing, they come later because you need more seed to be able to do them. But if we've got our selection funnel, through the selection funnel, you've already eliminated some of the potential variations. So the further down the funnel you go in your testing, the less potential you have for selecting hard. Within a single program, so if we think about a large commercial wheat breeding program, each breeder might make 500 to 1,000 crosses a year. Between those, that's 1 or 2 million plants. Each plant is different. Each plant is a potentially new variety. So they're screening up to 2 million potential new varieties in a single generation. And then as they move through the testing, they might take the best 5 or 10 varieties and put those into national list testing. The best 1 or 2 of those would eventually go into recommended list testing, and the best 1 of those might be recommended. Even then, there's no guarantee it will take market share. So there's a huge amount of selection that's going on, and it tapers off really quickly as the closer you get to market. I'll skip over this as quickly as I can. It looks complicated, but really it's about increasing genetic gain, which is the steepness of the improvement curve. You want crops to get better as quickly as possible. So this thing, delta G, is the genetic gain, and we want to do everything we can to make that as high as possible. We can do that by increasing the selection intensity, so by selecting as hard and as reliably as possible in the material we've got. We can do that by improving the heritability, so how accurate are the trials you're seeing? How sure can you be that your selections are actually the better part of the selections? And by increasing sigma A, so this is to do with the genetic variation. So the wider your genetic variation, the more chance you have of actually identifying something novel and better. So those are the 3 aspects you want to increase. You also, if possible, want to decrease the length of generation time. You want to go as quick as possible from making a new cross to taking the product of that and using it in the next cross. That's the way to steepen the curve and get the maximum genetic gain. In terms of technical limitations, then, what we want are large, diverse populations screened in accurate, uniform field experiments to get our best, quickest gains. And of course, regen ag situations kind of don't really fit this. It's hard to establish minimum tillage system trials and nurseries. So I think fundamentally we're selecting on different soils. If the tillage type almost goes half the way of determining the soil type, we can't do small plot work with minimum tillage equipment. Small trials and nursery plots are best drilled into a classic fine tilth. You plough it, you power harrow it, and then you take your small light kit, your agile kit, with a small amount of seed, and you can get some nice plots. These minimum tillage farm drills right outside here, they work by being large, efficient, heavy, and they move very, very quickly. You can't grow small plots with big bits of kit. It just doesn't work. So we need some clever engineers to come up and design us some minimum tillage plot drills. So if anyone's got a great idea, come and talk to me afterwards. Reducing input side of things, that's relatively straightforward. It's not difficult to have trials with lower inputs, but they tend to be less accurate. So you're more prone to gradients in the field of fertility in the soil, or the impact of pests in one corner can actually take out more of your trial. The weed burden might be uneven and this sort of thing, which means you might need to build in more replication, a better trial design, a smarter trial design. You might need more locations. Inevitably, if you have more locations and more years, that's going to slow down the rate of gathering enough good data to make decisions. So it might slow up our desired improved genetic gain. Then there are other questions. So we're hearing a lot about blends and populations, intercropping, things like that. How do we select for performance of components of blends other than by making lots and lots and lots of blends and testing them all? It's the numbers soon become quite difficult. So we need to think theoretically about how best to do that. Other systems like cover crops, undersowing, living mulches, grazing, how do we design varieties that are robust enough to do that early enough in the breeding program? I'm not saying I've got the answers. I'm saying these are technical reasons why breeding for regen ag could be difficult. Then commercially, as I've said, if the varieties are developed slower, this might lead to increased seed prices. So if it's taking longer to get a variety to market, the breeder's going to want probably more money back for that. If we're reducing inputs, does this mean we're having lower yielding seed crops? Again, that might impact on the cost of seed increasing. If the RL selection criteria change, and they have done in the past, it's only fair that there's then a kind of a burn-in period, allowing the breeders a chance to adapt to those new standards and let those new traits filter through onto the marketplace. Or, as one of our board members suggests, perhaps we need a conventional RL and a regen RL, but how on earth can we pay for two parallel testing systems? Then we need to think about certification standards and the royalty-based collection. So how would that need to change? Could we trim some of these costs away if we reduce our standards slightly? But part of the reason of the standards is it's helping to protect the intellectual property for the breeder and also to make sure that the farmer's got the best possible seed. Variety blends and populations are potentially valuable to help manage the risk to the grower. But how do they work with current royalty models? The blend that you harvest isn't the same as the blend that you plant, because as interplant competition, it will have shifted towards one type or another during the season, depending on what the pulls and pushes of that season are. So how do you then claim royalty on farm safe seed when the population, you don't know what the makeup of that is? And if we're talking about reducing inputs, are the end users we're selling the crops to prepared to take produce with perhaps lower specification? If we're reducing nitrogen, it's going to be harder and harder to make current 13% milling spec for nitrogen. So are millers and bakers prepared to reduce their standards to accommodate this? So these are all questions that I think need asking. So steps in the right direction then. Well, the public sector is definitely funding more crop research than 20 or 30 years ago, largely through BBSLC. Most of this is targeting the fundamentals of how to reduce inputs one way or another. We know much more about aspects of soil health, roots, and the microbiome. Every agricultural show you go to now has at least one soil pit. I've seen at least two here today. And most recently, DEFRA have an increased research profile. So their requirements seem to be much closer to what's needed for regen ag. So several opportunities are live, and there's an ongoing stream of opportunities at the moment, which is positive. Pre-breeding research has been a lot. This is to do with moving trait discovery from the lab into well-adapted material to pass on to breeders, and NARB have been very active in that. There have been several multi-partner projects since 2010. There is stakeholder involvement, though historically that's tended to be through breeders rather than through growers or processors. It's starting now to deliver pest and disease resistance, sustainable yield improvements, improved nutrition, and so on. And it's now starting to trickle through into breeders' germplasm. I've put up DSV's logo. They have a variety in National List 2, which is bred from some of our pre-breeding material in NIAB. So this is now starting to come through. But we need similar initiatives in other crops. This has happened in wheat, and it's happened in wheat only. It needs to happen in all the other grain cereals, in grain legumes, oilseeds, forage, and fibre crops. It can't just be isolated in wheat. And crucially, we need long-term experimental support. So the work we do at NIAB and the work that's done at Rothamsted tends to be funded kind of internally or by agricultural charities. It's not sexy science for the research authorities to fund normally. Very quickly then, some of the highlights from NIAB research. I had a trial a few years ago where I added no nitrogen at all. It followed winter beans. Most of it looked like the spreader had not worked, and then occasionally you stumble across a plot that's bright, vivid green. There's a reason behind that. There's obviously something it's better at scavenging for the nitrogen or relocating the nitrogen further in the season. Those are interesting leads for further crossing for low input. With some French funding, we've done work on a trait called grain protein deviation. So this is the negative relationship between yield and protein. What we're looking for is varieties that sit above the curve. They have higher protein than you predict from their yield. They have higher yield than you predict from their protein. And it's known that there are varieties that consistently do that. So breeders are interested in incorporating that into their material. And I had an interesting trial in 2019-20. So it was drilled far too late because the autumn was awful. It was drilled into a shocking seed bed with no blackgrass control at all, partly because the autumn was so awful and partly because it hadn't been controlled in the previous crop. And then, of course, we had a really, really dry spring with no rain at all. So it started, and I'd already said I only wanted 70% of the nitrogen putting on the trial. So it was designed to be a low nitrogen trial. It was drilled too late. It had a high weed pressure and it had a drought. It had pretty much everything thrown at it. But we're still able to pull apart consistent differences between varieties. So some of them were absolutely shocking and some of them were still outperforming the best commercial checks. So only by testing in crap conditions, I think, can you start to skew your material to perform well under low inputs. And we just need to be brave sometimes about not necessarily putting trials in the most perfect uniform fields. I asked for some input from HDB, and Paul Gosling is here to help answer any questions. And he came up with these, so thank you, Paul, for those. Since 2015, the recommended list has put much more emphasis on disease resistance. It's not just about treated yield. So, for example, light leaf spot has been raised in importance for the oilseed rape list, mildew for winter barley, yellow rust and mildew for winter wheat. There are special categories instigated now where there's a new trait of value to growers. So the first time this happened was with orange wheat blossomage resistance. The first variety, I think, was Welford, back when I was still breeding. More recently, turnip virus resistance, clearfield traits in oilseed rape, the Wolverine resistance to BYDV in wheat, and BYDV tolerance in winter barley. So there's a special class you can go on the list. It's not just about yield anymore. They've instigated a watch list for wheat yellow rust, so varieties that are potentially at risk of the ratings crumbling quite quickly. This is based on input from the samples that get sent in. They've put together a variety blends tool to help growers with designing blends from recommended list varieties. They're doing data analysis on the interactions between different varieties and the tillage system, so whether it was min-tilled, ploughed, etc. And they're enhancing digital access to the OL to help with all of these decision and variety selection tools. And finally, commercially, well, as I said, breeders are actively using pre-breeding material, so the broader genetics is starting to find its way into varieties. So these are just the breeders I know work with on designing future wheat. Seed companies are tailing their offers a bit. We're seeing a lot of blends. We're seeing a lot of cover crops, companion crops, specialist crops. There's no shortage of companies offering biostimulants, etc. So all the little, maybe 1-2% tweaks, lots of people are offering those. There's lots of kit and lots of advice available out here. And of course, the current situation with crop prices and input costs are making everyone think really carefully about what their inputs are and what their outputs might be, what their gross margins are, how's the best way to go about that. So I think growers are much more open to things than they used to be. So without saying anything else, I'll pass over to Stéphanie and we'll take any questions towards the end. Thank you.
Thank you, Phil. Hello, everyone. Hope you can hear me properly. So I'm Stéphanie Swarbreck. I'm a plant biologist. And I wanted to talk to you about our work to try to establish nitrogen responsiveness as a trait that can use to select varieties. So nitrogen is very often a macronutrient that's limiting in many different ecosystems. And you can see it very clearly. You can see on the photo here on the left-hand side, the plants are shorter. They are not as there's not as many tillers that have emerged, and they are not as green. So clearly, adding nitrogen kind of has a really strong effect on the architecture of the plant itself, on its capacity to fix CO2. And of course, we've been solving this issue of the lack of nitrogen in different environments by applying synthetic nitrogen fertilizer. And the application of synthetic nitrogen fertilizer has really led to and driven our yield increase over the last century or so. However, there are many downsides to applying nitrogen fertilizer. So the processes that is used to generate this nitrogen fertilizer emit lots of CO2. The application itself is really inefficient, so the nitrogen use efficiency ratio that you can get is really low. And the nitrogen, so because the ratio of uptake is really low, you get nitrogen that leach into the environments or N2O that is emitted. So clearly there's huge, it's really necessary to decrease our reliance of nitrogen fertilizer application. So really the challenge is to find a balance between supplying sufficient amount of nitrogen, because we still need to be able to supply sufficiently to produce the high yield, and the demand of the plants to make sure that the canopy structure and the plant itself is large enough, there's enough grain being produced, and that the grain protein content is high enough. So it's finding that fine balance. And if we don't find that fine balance, then we have issues with runoff and pollution. So a lot of the time we talked about nitrogen use efficiency. That's a term I've just used as well. So this is defined as the ratio, so grain produced per unit of nitrogen supplied. And this was kind of an easy definition to some extent. It makes sense to understand if you start thinking about the nitrogen that's taken up, where it ends up. It really makes sense. However, it's also kind of a problematic because it's so tied up with the yield. So as soon as you increase the yield for whatever other reason, maybe the architecture or the harvest index is different, then you're leading to an increase in nitrogen use efficiency. But have you really improved the processes that are undertaken by the plants? Perhaps not. So my thinking really started to change when we had discussions as part of a previous project, and we looked at this type of graphs, which I'm sure many of you have seen before, where we have on the x-axis and the bottom axis the amount of nitrogen applied, and on the side axis the grain yield. And as you would expect, if you supply more nitrogen, you increase the yield. And then if you supply even more nitrogen, at some point, you don't increase the yield anymore because you're reaching a plateau. And I think some of you might be familiar with the concept of this economic N optimum. So the optimum amount of nitrogen that you need to apply to maximize the yield. So beyond that point, you don't make that much profit because you've applied too much nitrogen, you haven't recouped it in terms of the yield. And then below that point, you haven't reached the yield potential, so you also haven't made that much profit. So really, it's a sweet spot. That's economic nitrogen, economic and optimum. So, but there are issues with this as well in terms of, because it's just one kind of a point. Also, for us as scientists, it's a bit more difficult to take into account the economic aspect. However, a variety that will have a low and economic optimum is also one that will be highly sensitive and highly responsive to nitrogen. So this is where hopefully some of you can see the green line is kind of shifted towards on this side, basically. So a variety that will have low economic and optimum will be responding and producing, growing better under lower levels of nitrogen, but also hopefully continue to grow and continue to respond to this added nitrogen even further, even when others might plateau. So I call this nitrogen responsiveness. And for us, it's quite important, I think, in terms of thinking of how we can select this variety that might be better. So I'll talk through some of our analysis of a set of data that was obtained and generated through the regen network. So they've tested 45 different varieties under 4 levels of nitrogen. They had a 5th one for 1 year only. And this was done over 15 years. So it's a really long-term experiment. And it's quite rare to have this type of data set, so it's quite interesting. And you can see the data that was plotted for the entire data set, so all the varieties for all of the years. And it's quite nice to see if we add nitrogen, yes, you increase the yield. But I'd like to draw your attention to these violin plots, and you can see how they are kind of elongated. And in some cases, the yield that you can see under really high, under low levels of nitrogen are kind of almost the same as the one for some of the points that are achieved under high level of nitrogen. And that's really what I want to get a handle on. So I started to correlate to see whether the yield under low levels of nitrogen was really an indication of the yield that you could see under higher level of nitrogen. And yes, it's positively correlated. So yes, to some level, when you grow the plants under low level of nitrogen, it can give you an idea of what yield you're going to get under higher level of nitrogen. But this really correlation really breaks down, and it's much less significant the further apart the level of nitrogen they are. So you can really get, so if you test your plants under 100 kilogram nitrogen, it will be a good indication of what you might achieve under 200 kilogram nitrogen. If they are tested under 0, it might be less of a informative. So if we start looking at what it means to have this correlation, here I've put on this slide, so each of this triangle here corresponds to 1 year, 1 variety, and it's the yield under 100 kilogram nitrogen and the yield under 200 kilogram nitrogen. So as you can see, as you increase, as the yield increase under 100 kilogram nitrogen, it's also quite high for the 200 kilogram nitrogen. And you can see it's kind of a positive correlation. Okay, that's important. I'd like to draw your attention to these data points here. And you might think, okay, that's just an outlier. Maybe that year in that plot and that particular varieties, there was, for some reason, disease, and that's why we don't have a very good yield at 200 kilogram, but at 100 kilogram, it was okay. Fair enough, but look at all of these points here. And the problem is that if we have plants that are grown under those conditions, and they are not necessarily gaining in yield under the added nitrogen, then we do end up with environmental issue and cost for the farmer. So how do we get about, and what do we do about it, and how can we get a better understanding of it? So that difference there, this is what I'm calling the yield nitrogen responsiveness. As you can imagine, is really dependent on environmental conditions. These are all of the nitrogen responsiveness that I could calculate over the different years. And 2008 in particular was really dry. It's really low. Okay. It was kind of expected that there would be some level of environmental regulation in that. But the Regen data set was really interesting because we could start testing whether there was a genetic component, whether those varieties that were responding better were consistently responding better. And what we found that Phil has already mentioned, this heritability. We found a heritability of 0.74, which is actually really good. And we could find some significant differences amongst those varieties in a set of varieties that we've been tested. So this is really good because we can start now drilling down into what are the genes, what are the part of the genome that are relevant for that specific trait. So far, we couldn't see any specific difference in terms of whether the varieties were from specific NABIM group or whether they were registered more recently or not. So we've used this approach before in a completely different species, in millet, and we've done genetic, and we've been able to identify specific area of the genome that would not be highlighted when we just looked at the yield. So looking at this nitrogen responsiveness, this index, has been already a useful approach. And where this is leading, well, at the moment, we don't have any information, or farmers and growers don't have any information about nitrogen, or the requirements of nitrogen, except if we're growing specific milling wheat where added nitrogen might be necessary for to achieve the grain protein content, for example. So having this kind of information, I think, is really important. And I've put there as well the competitiveness against wheat. There was other traits that would be relevant in there. But just like before finishing, I'd just like to mention one of our projects that started very soon in the autumn. It's with my collaborator, Nathan Morris, who is at NARIAB, and it's funded through the Morley Agriculture Foundations. And this is quite exciting because what Phil was just mentioning is that breeder test material under regenerative agricultural conditions really later on in the development of this variety. Well, at NIAB, we have a wealth of pre-breeding materials. We also have a wealth of try and really long-term try. And with this project, we're putting those together. So we'll be testing some of this pre-breeding material under this regenerative agriculture. And we're really hoping to get to a point where when we're adding new criteria, yes, there'll be yield we'll be looking at, there will be the national listing criteria, but what about environmental grading? Can we think about nitrogen responsiveness, as I mentioned, competitiveness against weed, how much emission can be can be produced from these different varieties? There's a lot more that can be done. And so this is quite exciting time for us. And so just to summarize everything, I hope I've convinced you that nitrogen responsiveness is quite important to consider, and that we can now have the tool to start exploring it a little bit more. And I'll stop there. Thank you.
So hello, good afternoon everyone. I hope you can hear me well. So I'm Ambrogio Costanzo, leading the crops team at Organic Research Center. I'm an agronomist, and we are a small team of agronomists, and we have played with wheat varieties, and now we are also playing with oats varieties, and hopefully in the future with bean varieties, a lot of other things, directly with the farmers. And that's just going to briefly talk about some of this evidence today with you. Can some of you just tell me when 2 minutes left, because we have all started a bit later. Just to start, share a reflection. Most of you will have heard about the agroecological transition. So how do we go from industrial agriculture, which luckily we don't have anymore, at least we don't have in this agricultural show, towards a system redesign? And we knew that if we start from farm as a factory and try to get towards conceiving the farm as an ecosystem, how does this link to seeds and plant breeding? In a way, it's nice to be the third speaking today, because this summarizes a lot of things that who preceded me have said. We started in history from 50, 60 years ago from varieties that were bred to maximize the responsiveness, so high input to get high output. And then we went towards getting high output with reduced inputs. But ultimately, we will need to get, so yes, to a situation in which seeds are in a way part of the ecosystem. So in all this transition, and we can see today in our agricultural reality still a gradient of all these 4 steps. A lot of farms are still industrial agriculture, spraying and spreading nitrogen based on a calendar without really thinking about when is the right time. A lot of farms are on the other steps of the transition, and a lot of farms represented here are quite at the avant-garde of the system redesign. What you see, if you compare varieties on all these stages, I speak about an expanded scope of breeding. And in fact, what you see that 2 varieties that may perform very similar here, as long as you move towards a more sustainable, let's say sustainable, lower input system, the divergence between these varieties gets higher and higher. But in which direction do we need then to go? So I'm just sharing these funny charts that I've just drew a long time ago to explain the situation. When the two extremes. So in the really industrial system, big companies during the so-called Green Revolution had high-yielding varieties sold together with a package of fertilizers, herbicides, and pesticides. You had a variety of environments. Here I've just put a dummy fertility and water availability axis, but it's just a concept. And basically, drilling these varieties and using those inputs, you tended to pull all the environments towards those conditions that were suited to the individual varieties. The ideal of seeds as part of the ecosystem is that each environment has its own seed adapted to its own conditions and everything. Of course, it's a sort of a utopia, but the key message here is that perhaps one possible direction we need to go through with breeding and cropping systems and everything is going from a broad adaptation, so a variety that's a one-size-fits-all, towards specific adaptation, so in a way the right seed to the right farm. But how to get there? In a way, there are possible two efforts that are made. So if you think about a breeding process, so you start from generating diversity, the crosses Dr. Phil Howell spoke about earlier on, then you have the selection, then you have the testing, and ultimately you have the life cycle of a commercial seed. One thing that we can do is what our colleagues at NIAB have done is, of course, pushing from the start of the process, so enlarging the genetic pool, doing new crosses and all these things. What we did from the Organic Research Center was starting from the end, so pulling from the end, so testing varieties into real farms at a field scale with organic and regenerative farms, and see how these varieties really performed without having really been tested for those conditions. And this is a project called Live Wheat. It's been going on for 3 years plus 2 years beforehand, so we are now running the 5th year. It's a participatory work with a group of farmers represented by mostly the Organic Arable membership-based marketing group, who have organized a group of 20 plus farmers. Each one of them was provided with seeds. We decided the seeds together. They drilled the seed in a sort of a tramline strips. The research team went through the fields and assessed the crop vigor, cover, weeds, even the weed abundance and the diseases. Farmers measured the yield, and then we did quality analysis of the grain, and this for 5 years, according to a specific experimental design by which every farmer could actually drill a subset of varieties, but coordinated with the other farmers, we could test more than 25 different varieties. So some evidence from this project that may kind of start trying to tie some loose ends of this very uncertain discussion. Please bear in mind these are very interim results. The last season is now in progress, so things may change. Some may become stronger, some may disappear. But anyway, in terms of environments, we have considered a lot of things. So for example, the region where each of the 20 plus farms were, the soil texture, but also the weed pressure. As we said, we have farms from conventionally regenerative farms, relying on herbicides, very clean field, in inverted commas, to organic farms with very high weed pressure. And then there is also a different chapter that we are not talking about the different weed communities. One thing, if you have a field dominated by blackgrass, one thing is if you have a field dominated by poppies and charlock. It's quite different. And also, my dream in the future would be to see how can we choose variety based on which weed is dominating in your field. But this is dreams. We don't have time for that now. And then we have the environments and the unpredictable side. We've had 4 years so far and 1 in progress. And we started from 2018 with a historic drought in summer, a 2019 harvest year I'm speaking about, notoriously mild and wet. 2019-20, we were just mentioning earlier on the late sowing and everything, and the hugely cold and dry spring, and in 2000 dry spring but not cold, hot, and usually cold and dry spring in 2021, last year. And this year is another chapter again. So anyway, we have 4 scenarios. We don't know what next year is going to look like, but what we're trying to end up saying is that what if we look like one of those, and what would be the best case and worst case scenario? Anyway, performance. Putting all the past 4 years together, imagine an ideal an average farm in the average year. We have the yield and protein trade-off. So the varieties that have high yield have lower protein, the varieties that have low yield and higher protein. And we see a cluster of feed varieties on the southeast, a cluster of 10 million varieties on the northwest of the chart. But Phil and Stéphanie were speaking about the grain protein deviation. And here we have a demonstration of what it means. If you take this chart for each of the 4 years and you try to understand whether one variety was consistently above the line, and yes, we found one, and this one is Maris Wigeon. So it's a variety, it's a historic variety bred in 1964 when the whole plant breeding progress was still the domain of the public powers and the public organization. And in organic and regenerative farmers, in 4 years, it consistently showed to the best, we could say, broadly speaking, nitrogen use efficiency, of course, with a much broader definition than that, but still more protein with still a decent yield without having a very good very market trade-off. And then we looked at the yield and protein, also, of course, how big they are, but also how consistent they are. And these are quite interesting charts to look at, especially when we look at performance across farms. It's called dynamic stability. If you put all the farms in every year on the horizontal axis from the lowest yielding to the highest yielding, we have a control variety that is the dotted line, and we want to see whether what happens when a variety, when we go to high yield environment or low yield environment. And what we see that in terms of yield, we have 2 specific varieties in inverted commas that show better stability, consistency. One, the one you see above, is a blend of Siskin and Extase. It's interesting to know that this is a very late variety and a quite late variety and a quite early variety. And this showed higher yield, lower protein than average. And the yield, but you see that the yield advantage compared to the control variety gets bigger and bigger as you go towards lower yielding environment. And then we have YQ-CCP, one variety that you can see as a population. If you make a cross and instead of selecting one single variety from the progeny, you just grow the progeny, and then you end up with a population. This actually is the children of 20 crosses, 20 marriages altogether. Again, lower yield, but higher protein. This is not a big deal. The thing is that the yield disadvantage gets lower and lower as long as you go towards lower yielding environments. So these are 2, let's say, seeds, one variety mixture and one populations that are more stable, more consistent. Same thing we see with protein, with the addition of one specific variety called Extase. Thank you. And what about weeds? We were speaking about weeds. We have a lot of organic farms in our network. Here you see on the horizontal axis, as you move to the right, I struggle with right and left, you move towards higher more weedier environments. And if you look in the springtime, so when vegetative development, we see that, again, the composite cross population, the mixture and the variety Extase have lower weed abundance than the average. And if you look at them at what would be nowadays, mid-June, late June, you would see Maris Wigeon, which you will remember be the grain protein deviation, to be the one with significantly lower weeds. Pictures speak by themselves. Please note in the picture as stem extension, what you see as a bigger crop is not a bigger crop. This is wild oats. So actually, you see what a huge difference variety choice with just what's on the market, without going and look at the pre-breeding, just the variety choice of the market can make if you're struggling with weeds. Of course, we don't know it until we try that. And this is, of course, one of the classic pictures with poppies, with the Maris Wigeon on one side and the modern varieties on the other side. So, in discussion, I'm trying to hear, just to conclude, yes, there are 3 things here. There are no silver bullets, but there is something that we can look at, because we call it the functional approach. So what's meaningful for what we expect? Early vigor, so vigor during winter, during the vegetative stage, is very important because it reduces weed abundance, the so-called critical period for weed competition, and it ends up enhancing yield consistency. We saw it with variety X days, for example. Light early vigor, so the straw length, of course, it reduces weed growth later on in the cycle, and it can lead to a better nitrogen use efficiency. But the key thing here is that, especially in organic farms, and for whichever conventional regenerative farms that sooner or later will end up giving up herbicides, a lot of nitrogen use efficiency will be strongly linked to weed competitive ability, and probably going to be the same thing. And one day, sooner or later, we'll all need to give up or reduce drastically the herbicides as we are now reducing nitrogen. And on the other side, can we rely on diversity for resilience? Yes, and two strategies. One is blends. We have some blends out there, but one thing about blends is to really focus on the functional composition. A lot of blends now are done by trying to reduce, to increase disease resistance, but there is more than that. If you mix a variety with a late cycle and a variety with an early cycle, you minimize the competition between the 50% of each. And we've achieved, as we saw earlier on in the chart, a massive overyielding. And then we have the functional diversity of these populations, that if you make the crosses that Phil was speaking about, and instead of trying to select the best silver bullet into the millions of progenies that you have, you just grow this crowd of children all together, and imagine that you grow half of it on a no-till, half of it on a tilled environment, half of it in presence of weeds or perhaps on a clover understory. And every year, as long as there is going to be heterozygosis and there's going to be segregation, new plants and new diversity is being generated, you have a shortcut to that famous specific adaptation, because you're going to have three completely different populations. One adapted to high weed presence, which can be simulated by clover understory, one adapted to no-till. And this means bringing back the breeding into the farmers' fields. And of course, breeders may have huge opportunities in this, but may need to think about changing a little bit the business models about breeding. I hope I haven't been so long, but in conclusion, yes, the best way forward is still keep testing all together. Farmers keep testing varieties, link up and collaborate. And thank you very much for your attention.
Right, it's a bit loud. Thank you, everyone. Very happy to take questions about the talks, or if you have any other questions you want to raise, then the three of us will do our best to try and answer those. So who wants to start? Yeah.
Hi, thank you very much for the talks. You were talking about the challenges with long breeding timelines. With new breeding technology being allowed in trials now, like CRISPR, do you think this will be moving into commercial breeding anytime soon?
Shall I start? I'm sure. I suspect I will differ from Ambrogio on my thoughts about this. So, I'd be very surprised if the commercial companies didn't enter into CRISPR. How quickly they do that kind of remains to be seen. I know that some of the research organisations, so at NIAB, we're already doing CRISPR for experimental purposes, and we're talking to breeding companies and others to do that, similar at John Innes, similar at Rothamsted, and so on. Personally, I think CRISPR is another exciting tool, and we will need all the tools in the box, but clearly that doesn't suit everyone, and ultimately I think the market may well decide quite how we segregate and regulate and so on. That's a big discussion that we need to have, but we need to have that discussion with everyone rather than think we've got the best solution ourselves and try and impose it on people, I think. Ambo, do you want to?
But I think, yes. Of course, I have a sort of fidelity to the organic movement, which of course doesn't look very enthusiastically about genetics. But the only real doubt that I have, because we do need to have all tools in the box, but we also need to know how they are used. I just think that it can be very useful, but it's definitely not going to be, someone still needs to prove me that it's going to go in the direction of specific adaptation. I mean, I think that just making the accounts of how much the intensity of investment goes on there, going to reduce costs, but you're not going to have, we keep going to have better, much better and much faster, but still silver bullets. So they will work, we will have traits that we wouldn't even suspect have, but we won't have the diversity probably we need. So yes, it might have a place. Organic is a tiny percentage here, slightly bigger percentage in France and other places. So the problem is, I don't think it's going to be a long-term game changer. It may be a game changer on the market and on the balance of power, but this is another topic.
Yeah, I was just going to add, really, basically, is what it's to consider the traits that we can modify for it. And with the CRISPR, you can just modify one single gene. And a lot of the traits we've been talking about today, like the nitrogen responsiveness or that the competitiveness against wheat, there's still many genes that are involved and underlying. So we might be able to modify some specific aspect of it, but to go from that to a variety that is really high yielding, that's well adapted, and etc. It's just a long way. But it's really exciting to have this possibility, definitely.
Tom at the front.
Thank you for that. Tom Allen Stevens, Oxfordshire farmer, leader of the British On-Farm Innovation Network. Just a quick observation, firstly, about the gene editing thing, first of all. I mean, I've been looking at the programs that are going on at the moment in those, and it's very interesting to see that some of the work that's going on is looking at knocking out genes to see what they do. And then once you've done that, you've got the genetic markers, and there's no reason why you can't then conventionally breed wheat that can be used in organic systems, even if it's, if you've just got the information that's been generated by gene editing. Secondly, and my question really is certainly Phil and Ambrose, one of the things that you brought up was the lack of granularity of data and the sort of breadth and of data. I mean, what opportunities are there for the farming community itself to add to that data? For more on-farm research, better coordinated, and more scientifically coordinated. A lot of the recommended lists work. Could that be, and bring in different systems and so on and so forth? How could the on-farm research network add to the data that you currently get in small plot trials?
Yeah, just to say that actually live wheat as is very much farmer-led, and especially is very much field scale. So real commercial farms, so the farmers get the seed or provide the seed themselves. They harvest, they measure the harvest, and then they sell the grain as they would sell anything else. This is real data that is crucial to validate what we as a scientific community, we Organic Research Center, we have 2 feet in 2 different shoes, and as a scientific community, we get from the small plots. And getting this interplay between the 2 scales is absolutely crucial. We hope that live wheat, besides providing some nice charts about the stability of one variety or the others, can be a stepping stone into coordinating farmers with one another, because that's a crucial thing. Work as a collective, not work as individuals, to create the robustness of knowledge that we get, to make it work for many of the challenges as well. And there are other companies also here on stand today. They are providing some ICT tools, for example, to collect data and share data together and see what we can do. And I think my scientists, Half, and my colleagues here would value enormously what we can have from using the farms are actually experiments. The thing is actually, are we going to want to work as a community all together with actually the farmers and the breeders and the enterprises? And there is quite a bit of, we're all willing to do that, but there are a bit of obstacles that we are not guilty of that we'll need to overcome for that.
I think, so one of the reasons we don't do small plot work because we like working on small plots. We do small plot work because sometimes there's literally a handful of seeds in the world, and that's all we can do with it. But if we were to deal with as many potential varieties on a larger scale, the maths just becomes impossible the seed multiplication, the costs in doing the seed multiplication to give you enough seed to run your slug-resistant trials or whatever. It's fine for a few lines, but the power in selection comes from having lots of things to choose from. And I'm sure there's a trade-off, and I'd love to talk more about it in the future, but unfortunately, we need a mechanism to pay for that. So not just for the testing, but for scaling up the seed multiplication to the point where we can do the testing. And if there's no product to sell, if it's just to test a hypothesis about a parent that then goes into a breeding program, then again, how does it all get paid for in the end? I'd love that perhaps there was a government institute that could pay for it, but I don't think that's going to happen.
We have DEFRA supporting LiveWheat, though.
Yeah.
So
there's possibly some support, and certainly the waves of DEFRA funding that are coming, there's an awful lot of kind of co-design expected in them, and a lot of engagement with growers, far more than BBSRC expect. So I think we just need to pick our battles wisely.
If I can add just one tiny thing. From the field trials, of course, we can get something, but the results are not saying you grow X days or YQ and you're going to double your yield. What it's going to say, perhaps, is if you pick the varieties that have better early vigor, you are more likely to have a chance to are going to succeed in organic farms, for example. But then the early vigor, you can do it. You can measure it even in a pot, as long as you don't pump 200 kilograms of nitrogen per square meter, is going to be pretty reliable. So this is the interplay. When you have if the big field can inform what you can do when you have a bunch of seeds.
Early vigor is interesting, actually. I saw a chat somewhere on Twitter yesterday with someone suggesting that early vigor seems to be a key trait for establishment in reduced tillage situations and so on. So varieties that you would normally put in a later drilled situation, they need that early vigor. They seem to do better under reduced tillage compared to a slow developing early sowing variety in wheat. This is, but so we need to listen to feedback from people about what suits their system, and if we understand what traits are involved in solving the problems of their system, then we can go towards selecting on those traits without necessarily having to do the testing on their farm all the way through. So there are potential ways of doing it that way as well.
Hi there, Tom Matthews, Dorset Somerset Border Farming. Obviously, your research has been off nitrogen rates in the winter wheat, but have you looked into the micronutrients and lack of or over availability and the effect on yield as a result of those?
So we do general soil mineral nutrients analysis with. I think normally the trials are supplemented with some of the macronutrients, and we haven't looked specifically into micronutrients and how they could be. I don't think necessarily for the nitrogen may be important. The phosphorus for sure. Like iron deficiency sometimes is a way of for the plants to show phosphorus deficiency or something. But yeah, we're focusing on one. I think as scientists, sometimes we have to simplify the problem so we can find a solution for that aspect of it. Yeah, thanks.
It's probably a piece which was initially touched on. You're looking at low input or difficult situations, traits, how that's coming across. And one thing that's always been difficult in breeding is in regenerative ag, reduced tillage, is probably rooting phenotypes and how do you phenotype in a solid mass. And going forward, how are you doing that? Is that important, more increasingly important in a scenario which might need it more? And then on top of that is soil interaction with the microbiome and mycorrhizal interactions, because there's a big talk about some varieties having more interactions than other, and that might make nutrient availability at a wider scale. Just really what your thoughts on that? Is that being explored, or is that too difficult to upscale on a sort of breeding timeline?
Oops. We are looking at root traits, as you said, they're difficult. So we've had colleagues at NIAB who have been part of big international projects. I vividly remember last year a trial where they had 2 plots side by side of each variety, taken right through to maturity, and then the day before the yield plot was combined, the next door plot, they were driving coring machines in to take samples. So it takes a long time to take a coring sample. It takes even longer to wash it out and do the analysis on the roots at various points. So I don't know what the results from that are, but the idea is that they're grown side by side, so you have the correlation between the yield performance, you have the grain quality that comes off the grain sample, and then you have the roots to go alongside it. We have done some work as well on some high throughput screening. So there's something called a clear pot screen. So you find a plant pot that's clear plastic, you slot that inside a normal pot, you grow your seedlings, and then you take the clear pot out and you photograph the seed roots. So there are some hypotheses about early rooting. There's this idea, it came from Australia, so I have to do the accent. You have a steep, deep and cheap system that goes down very quickly and gets moisture from depth as quickly as possible. So in certain scenarios, that seems to be the archetype. But different systems will suit different situations. So, Stéphanie, you've done work on roots, so.
Yeah, I think you've summarized quite a few. It's very difficult to do it, and even as a scientist, when we look at roots, there's always this difference between if you want to go really high throughput or if you want to be going and have lots of information. So we have system where you can take images of roots and it takes an hour to count all of the roots. But it's really important because you can really understand what's happening, and you can measure, for example, the laterals or the very fine roots that are important there. So I think Phil mentioned that there is another high throughput method. There's like a germination paper where you can also grow the plants to germinate your seeds on this paper, and then you can look at the root system architecture. Those type of root system architecture, they tend to be much more simpler to look at because you don't have all of the heterogeneity of the soil. But it gives information on the genetic aspects. I think in the last 10 years or so, scientists and breeders have come to the idea that we need to look below the soil and see how we can improve that. And there's lots of differences. So the work that I have done so far has been looking at the interaction between weed and wheat and trying to see whether the presence of a weed will be affecting the root system architecture with black grass in particular. Some of the growers were telling me that what really matters is what's happening below ground and not so much about above ground interactions. And we could see that if a wheat plant is grown in the presence of a non-wheat, so in the presence of a neighbor, the lateral roots are much shorter, so it's less of a capacity to take up nutrients. So yeah, there's clearly some work to be done there.
Louise Payton from the Soil Association. My understanding is then that the biggest kind of barrier to having the right kind of traits available for regenerative agriculture and organic agriculture is that you're dealing with low chemical use environments which require more local varieties. It's based on what you were talking about, Stéphanie, in terms of high chemical systems tend to pull all the different varieties towards the same kind of outcome. So, and at the moment, it sounds like the structure of breeding is not geared towards that. It's very much more about one-size-fits-all varieties. What is the main... At the moment, the government is obsessed with gene editing being the answer, but it feels like there's something... I guess I'm particularly interested because I work in policy. What should the ask be for government for how to support breeding towards these kind of things?
I was going to say the recommended list. I was going to say that we keep going back to that. I've got loads of conversation about the nitrogen responsiveness work that we do and the weed competitiveness project. And then breeders tells me this is not something that are particularly interesting because it's not on the recommended list traits. So we might look at that later on and it might be useful for us to sell that particular varieties. But at the moment, this is not something that we'll be selecting for. So that's what I would say.
So perhaps if some of the burden were taken off the levy payers who, through the HDB, fund the recommended list, and the recommended list were able to be even bigger and take in far more diverse cropping situations, far more diverse inputs. We would only need one recommended list. It might be enormous, but there might be ways in which you could interact with that to find varieties that are suited to your particular situation, because there would have been enough testing in those situations to find the winners and losers. But I think it's unfair to expect the AHDB levy to cover that. The trials would have to be so enormous that you can't possibly do that with the current model. So perhaps we need a different model, and maybe the government could help with that. I've not thought of that before.
To go around that problem, which seems to be decades talking about, some experience from the European projects focusing on organic breeding and variety testing are the alternative business models. So perhaps you won't need too much of official things. There is a need to have more varieties for more diverse seeds for more diverse environments, and you have much less resources to test for it and to breed for it as well. So typical breeding now, the return on investment is based on how many hectares you cover with a variety. A variety has a cost of £1 or £2 million, for example, just shouting numbers from an old friend from one breeding company who told us that. So, of course, you need to cover quite a big surface. So, yes, specific adaptation is not East Anglia variety adapted to East Anglia and variety adapted to the West. It can be really about a specific tillage and specific wheat community. So one shortcut is up there, is the population potentially. And there is I don't know if I can already say that, but from a government department whose headquarters is not far from Westminster, I can say what it is, genuine interest to follow what is going on in the European Union with recognizing the legality of selling seeds of heterogeneous material. It's illegal. It's illegal because, of course, it's not the US, it's not distinct, uniform and stable. You can't apply intellectual property rights on it. Do we need to apply intellectual property rights or anything? Probably not. I mean, a lot of research from top Nobel Prize winner economists say that perhaps a lot of innovation especially if it's dealing with a public good, goes much faster and much wider reaching if you don't apply IPR. And then there are supply chains. We have had this stage hosted a fantastic session this morning on short supply chains and how they can sort of ensure return to also to potentially breeders. Consider that breeding a population costs much less than breeding a variety, ultimately. In the 60s, it was researchers from the US who started doing that and proved that with a very minimal effort, you can have an increase in yield. So, yeah, a bit of fantasy probably, and a bit of policy, not necessarily to improve the existing system, but also to allow what now is illegal, make it recognized, and we've done this. In the European Union has done it as soon as we have left, and now, of course, there is a genuine interest to see whether we can even take this as an opportunity to do it even better than in the European Union. Well, I don't know, but
so that would need a kind of a change in the whole seed system, not just in breeding. It would be the marketing and distribution and everything else along the way. So,
yes, a sort of a tracking system to make sure you know what you're growing, but you don't necessarily grow a population called Phil whilst you think you're growing a population called John, and making sure the seed safety. Generally, they have attached, the legal system attaches the seed health, the seed safety, so the seed has no bunt, has no weed seeds into it, with the identity, making sure that this variety is that Syngenta or KWS variety. If you just separate these two things, it maximize the attention on seed health, but just track to make sure you know what's what on the seed identity. I mean, there are a lot of pathways can open. Just make sure people don't get fined if they do it. That's as easy as that. Thanks.
Okay, I think we've really overrun. So
this is an example of breeding and selection from a huge population, getting the best of it. Yeah.
Thank you, everyone, and enjoy the rest of the day.